In sum, most animals do not have fully functional sensory systems at birth. Both normal development and the fine-tuning of sensory abilities depend on postnatal sensory experience. The remarkable changes in sensory function that occur with altered sensory input can provide clues to the evolution of sensory systems. Again, the visual system can be used to illustrate this point. Environments with poor visual conditions, such as caves, underground tunnels, or turbid waters, are often inhabited by animals with little or no visual acuity. Cave-dwelling salamanders are an excellent example. They have eyes at the larval, but not the adult stage, and consequently no vision. Amazingly, if these animals are reared in light conditions, they develop normal eyes and low-level vision. This suggests that the genetic code for building functional eyes is present in these animals, but is not expressed under their normal living conditions. It may be that vision provided some evolutionary advantage to the salamander's ancestors, but was no longer necessary when the animal's habitat changed. Perhaps vision remains viable in these animals because evolution has not selected against it. Like any other trait, if there is no disadvantage to vestigial vision, including that it does not require metabolic energy to be maintained, there may be no reason for it to disappear across evolution. Sensory system function. Sensory system function can be divided into two stages: detection and processing. Sensory detection describes how animals acquire information about their sensory world. Sensory processing describes how this information is organized and transmitted to other brain regions. Sensory detection and processing allow animals and humans to interpret the world around them and to respond appropriately to stimuli in their environment. For example, an animal that could not tell the difference between a mating call and an alarm call would be severely impaired in most social interactions. Sensory detection. Sensory detection begins at the sense organs: eyes, ears, nose, skin, tongue, which each contain groups of specialized neurons called sensory receptors. Sensory receptors transmit information to the central nervous system through a process of neuronal communication. Neurons vary in size and shape, but all have four primary features that are necessary for interneural communication. See Figure 2.8. The soma, or cell body, contains the cell nucleus and other structures that aid cellular metabolism. Neuronal signals that are used for communication are generated in the cell body and then transmitted by electrical impulses along the axon. This long, thin output fiber of the neuron has many branches, each of which swells at the tip to form a presynaptic terminal. When an electrical signal arrives at presynaptic terminals, it causes the neuron to release chemicals called neurotransmitters. Dopamine, serotonin, and acetylcholine are all well-known examples of neurotransmitters. This transmission of an electrical signal from the soma along the axon to the presynaptic terminal, resulting in the release of neurotransmitter, is called an action potential. In neuroscience terminology, neurons fire an action potential, and a neuron is firing if action potentials are generated. Figure 2.8. Neuronal structure showing the transmission of information within and between neurons. An action potential is generated when electrical signals within the neuron are transmitted from the cell body along the axon to the presynaptic terminals. This causes release of neurotransmitter, which acts on the postsynaptic neuron. Okay, so in this diagram you can see the presynaptic neuron. So it's got dendrites, these little tendrils. It has a cell body with a nucleus, and then it has a really long, stringy axon that has little branches at the end, and it does seem to swell out at the end. And then the electrical impulse flows from the cell body along the axon to the little nodes at the end. It's the direction of the action potential. Then you have the postsynaptic neuron beside it, and which has the nucleus and then all the dendrites, like the stringy branches around it, and some of them are next to the nodes at the end of the other one. So you can see that the first cell's axon terminals synapse with the dendrites on the target cell. So the chemical neurotransmitters pass between them from the axon with presynaptic terminals of the first cell to the dendrites of the second cell neuron. When a neurotransmitter is released from the presynaptic terminal, it crosses the synapse, a narrow gap between the axon tip of one neuron and the cell surface of another neuron, to act on postsynaptic receptors located on the soma and dendrites of other neurons. Dendrites are tiny fibers that branch out from the soma, increasing the surface area of a cell so that more information can be received from neighboring neurons. Activation of postsynaptic receptors causes biochemical changes in this neuron that increase or decrease the probability that it will fire an action potential. If the probability of neuronal firing is increased, the connection from the presynaptic to postsynaptic cell is excitatory. If the probability is decreased, the connection is inhibitory. A single neuron may receive input from dozens, hundreds, or even thousands of other neurons. The combined influence of all excitatory and inhibitory input determines whether an action potential will fire at any given moment. Sensory receptors are unique in that they respond not to neurotransmitter release, but to input from the environment. This input arrives as physical events such as sound waves, hearing, light waves, vision, or airborne chemicals, smell. When these events reach the sense organs, sensory receptors are activated, i.e., fire action potentials. These sensory receptors translate physical events into electrical signals, a process called transduction. In this way, sensory input is encoded as neuronal messages that are transmitted to other parts of the central nervous system, including the brain. Action potentials are all the same, so the brain must have some way to distinguish different types of sensory events. This is accomplished, first, at the level of the sensory receptors, which respond to a specific type of physical energy. For example, visual receptors in the eye are activated by light waves, auditory receptors in the ear are activated by sound waves, olfactory receptors in the nose are activated by airborne chemicals, etc. This specialization ensures that action potentials in the visual system are interpreted as light, whereas those in the auditory system are interpreted as sound. Gently pressing on closed eyelids produces an illusion of small dots of light because the brain interprets stimulation of sensory receptors in the eye as visual input. The effect is even more dramatic if stimulating electrodes are connected to nerve fibers that carry visual, auditory, or olfactory information. In these cases, individuals experience lights, sounds, and smells that are not present in the environment. Sensory processing. Sensory information remains segregated by modality as it is transmitted from the sense organs to higher brain regions. This segregation occurs because there are separate neuronal pathways for vision, audition, olfaction, and other senses. Each pathway begins with a group of sensory receptors that sends axons to the spinal cord or brainstem, where they connect to another distinct cluster of neurons. These neurons, in turn, connect to other groups of neurons, with each pathway terminating in a specific region of the cerebral cortex. Thus, each sensory modality has a distinct pathway from the sensory receptors, where information is detected, to the cortex, where it is processed. This ensures that visual, auditory, and tactile messages do not get scrambled as they are being transmitted. This principle is illustrated in Figure 2.9, which shows the sensory pathways for vision in humans. Over human evolution, the number of stops along this anatomical route from sensory detectors to higher brain regions has increased, allowing more complex processing of sensory information. Figure 2.9, neural pathway for transmission of visual information in the human brain. The visual projection pathway. Okay, so you have the left visual field in front of the eyes and the right visual field. Then you have the left eye and the right eye looking at a point in the visual field. Then you have the optic chiasm. Then you have the pretectal nucleus, the superior colliculus, and then it finally travels to the left cerebral hemisphere. And at the back of that is the visual cortex where it is processed. And this is all happening on the left side of the brain. And on the right side of the brain, where the message is not getting transmitted, you have the lateral geniculate nucleus of the thalamus and the right cerebral hemisphere. In the preceding figure showing sensory processing in the human brain, the visual cortex is much larger than the auditory cortex. This reflects a general principle of brain organization. The size of cortical area devoted to a function is a reflection of its importance. Humans rely much more heavily on vision than on audition, so we need a larger area of cortex to process this information. In contrast, animals that use other senses to gather important information about their environment should have larger cortical representations of these senses. See Figure 2.10. Figure 2.10. Examples of cortical magnification for A, the bill of the platypus, B, the nose tentacles of the star-nosed mole, C, the hand of the raccoon, and D, the whiskers of the rat. S1 equals sensory cortex. Okay, so it's cortical magnification. So you can see the duck-billed platypus, so he has a very big nose. Wow! About a third of his brain area is for his nose. With the star-nosed mole, you can see a decent size is devoted to the nose. And for the raccoon, about maybe an eighth is devoted to the hand. And for the rat, wow, it's a pretty big, maybe like a sixth or an eighth of the brain is devoted to the whiskers. Night hunting bats that use echolocation to identify prey have an exceptionally large auditory cortex. Other nocturnal animals, such as the platypus, Ornithorhynchus anatinus, spend much of their time searching through muddy waters for insects, shrimps, and other invertebrates. The platypus locates these prey by moving its large bill back and forth under the water. Sensory receptors on the bill detect mechanical ripples from other animals, indicating both their location and direction of movement. Accordingly, almost all of the sensory cortex of the platypus is devoted to the bill, with minimal representation of auditory and visual areas. Coding stimulus dimensions. It is clearly important for organisms to distinguish visual from auditory from olfactory sensations, but this provides only a crude snapshot of the sensory world around them. Animals must extrapolate more detailed information about sensory stimuli in their environment if they are to use these cues to locate prey, attract mates, and care for their young. Information that is likely to be important to animals in these situations includes the attributes or quality of the stimulus, the intensity of the stimulus, and the length of time that it remains in the environment. This section outlines how the sensory system is set up to detect these differences. Sensory quality refers to the type or category of stimulus within a modality. That is, different sounds, smells, or tactile sensations represent qualitative differences within a modality. These are coded in the central nervous system by the activation of different groups of receptors. For example, low and high-pitched musical notes activate different combinations of auditory neurons. Similarly, sweet and sour tastes are distinguishable because the chemical qualities of these two tastes trigger different sets of neurons. The same is true of the somatosensory system, with hot, cold, ticklish, and painful sensations producing action potentials in distinct sets of neurons. Sensory receptors often respond to more than one stimulus, i.e., taste receptors often respond to more than one chemical, and auditory receptors often respond to more than one sound, although stimuli that activate a single sensory receptor typically share certain features. That is, one neuron in the visual system may respond to different lines of a similar orientation, and a neuron in the olfactory system may respond to a number of chemicals of a similar structure. The nervous system, must also have a way to code the intensity of sensory stimuli so that organisms can detect differences between bright and dim lights, soft and loud sounds, and light and heavy touches to the skin. The first way that sensory intensity is coded is through the rate of cell firing. As the intensity of the physical stimulus increases, so does the frequency of action potentials. This principle is called frequency coding, and it explains how the nervous system distinguishes weak from strong sensory input. But neurons can only fire so quickly because the cell membrane must recover after each action potential before another electrical impulse can be generated. Because of this, a change in stimulus intensity beyond a certain threshold will not be detectable by single neurons. This problem is overcome by having more than one neuron fire at a time. According to the principle of population coding, as the stimulus intensity increases, the number of sensory receptors firing action potentials also increases. Stimulus intensity is coded within each modality, therefore, as an increased rate of cell firing and an increased number of firing neurons. The relationship between frequency and population coding is illustrated in Figure 2.11. Figure 2.11, intensity coding in sensory neurons. A. Each of the three nerve cells represented has a different threshold, low, medium, or high, and thus a different rate of firing. Each cell varies its response over a fraction of the total range of stimulus intensities. B. Although none of these nerve cells can respond faster than 150 times per second, the sum of all three can vary in response rate from 0 to 450 impulses per second, accurately indicating the intensity of the stimulus. Okay, so diagram A, on the x-axis you have stimulus intensity in arbitrary units from 0 to 20, and on the y-axis you have neural response rate, impulses per second, from 0 to 200. Okay, so it's showing three neurons. You have a low threshold neuron, a medium threshold neuron, and a high threshold neuron, and they can all fire at about 150 impulses per second. And then in B, on the x-axis, you have the stimulus intensity, arbitrary units, and on the y-axis, you have total neural response rate in impulses per second from 0 to 450. Oh, so you add the three responses together and it can reach all the way to 450 from 150 from the first one. Okay. So at the bottom, you see the responses of the low threshold neuron only go to 150. Then the combined responses of the low and medium threshold neurons go to 300. And then the combined responses of all three neurons reach 450 impulses per second. Coding stimulus duration. The intensity of a stimulus indicates how much physical energy is reaching the sensory receptors. By coding stimulus intensity, animals can detect how bright lights are, how loud sounds are, and how pungent smells are. It seems almost trivial to say that animals also need to know how long these stimuli last. A sudden burst of noise and a persistent roaring probably mean something different, even if they are the same volume. This leads to the question, how does the central nervous system code stimulus duration? The simplest solution may appear to be sustained firing. As long as a stimulus is present and does not change, action potential should continue at the same rate. The drawback to persistent cell firing is that it depletes energy and resources within the neuron. It may make more sense, at least metabolically, for the nervous system to respond to changes in sensory input. This also makes sense in terms of what animals need to know about their environment. Sights, sounds, and smells that remain constant probably have less significance than those that arrive or leave suddenly. The ability of animals and young children to keep track of temporal duration will be discussed in Chapter 6. Sensory receptors code whether stimuli are constant or changing through altered patterns of cell firing. If sensory input is constant, the rate of cell firing declines. This decline may be rapid or slow. Indeed, different groups of sensory receptors can be categorized as slowly or rapidly adapting. Slowly adapting neurons fire a burst of action potentials when a stimulus arrives, then continue to fire at a slower rate while the stimulus is present. Rapidly adapting neurons fire a quick succession of action potentials, both at the onset and the offset of a stimulus. The duration of a sensory stimulus, therefore, is represented in clusters of sensory neurons that exhibit different patterns of adaptation to persistent stimuli. The combination of these two firing patterns allows animals to distinguish sensory input that remains constant from that which is changing. For all organisms, including humans, sensory detection occurs automatically with little or no awareness of how this happens, but the process is far from simple. The segregation, coding, and organizing of sensory input provides information to the organism that is critical for survival. An animal that is unable to detect sensory information is unlikely to live, let alone reproduce and take care of its young. Beyond this basic level, sensory detection and processing are fundamental to all other cognitive processes. By coding specific aspects of the sensory environment, animals are able to find their way around, Chapter 5, make choices, Chapter 8, and communicate with other animals, Chapter 12.